I think AI is at the peak of its hype at this moment, and especially with the release of ChatGPT.
What are the qualities, if you have to trace back your younger self, what
Yes,
will you tell your 22-year-old self or 25-year-old self?
yes. My suggestion to all the bioinformaticians: learn how to code so you can judge the quality of the code. If you want to be really successful, I think you really need this self-discipline, I would say. That's actually one key thing. Life was tough, but then what it taught me is that you had to actually work really hard. One of the misconceptions is like doing analysis is very easy, like just press a button, then you generate the figures, like that's a one key misconception. Of course, AI can help, patient stratification, maybe site coordination to recruit patients. I hope now it takes 10 years, maybe it reduced to 8 years. That's still a very good, actually.
Hello, everyone. Welcome to a series of podcasts with people who help shape the modern world. Today, we have Tommy, who's a bioinformatics professional who works in AstraZeneca. Hello, Tommy. How are you doing today?
I'm good. Thank you very much, Akash. Thanks for having me.
Yeah. So I wanted to understand, since we are going through a hype cycle in AI, how does bioinformatics profession or biotech in general get affected by this hype cycle, and how do you see these two fields converging from your standpoint?
Yeah, that's a great question. I think AI is at the peak of its hype at this moment, and especially with the release of ChatGPT, so many people just adopt it, like millions of people, like in 8 months, something like that. It's like very fast. I think it's very good at many tasks. For example, summarizing, content, do deep research, for example. And for bioinformatics, those pharma companies, the biotech companies, they're also trying to adopt it to, for example, speed up the drug development process. And for bioinformatics specifically, we are primarily using, for example, GitHub Copilot or Claude, just to write better code. So that's actually like the primary thing that I see, at least in my daily job. And I think AI is great, in writing code, but also my suggestion to all the bioinformaticians are still lay a really solid foundation, learn how to code so you can judge the quality of the code, because sometimes give you just like good code but the wrong answers. It's very often, yeah.
So I would say, if I have to understand it from a layperson who is not from bioinformatics, bioinformatics, the core skill set is shell scripting, Python scripting,
Right.
applied to bigger data sets. Is that a fair summary?
I think that's pretty close. People actually use different terms, like sometimes for the same thing. For example, computational biology versus bioinformatics. To be honest, people use them like interchangeably.
Okay, but you would think computational biology and bioinformatics are synonymous in your point of view?
Yes. I used to think they're different, but it turns out people think the opposite, meaning someone's computational biology is someone else bioinformatics. So I just feel like it's not helpful to distinguish them. And initially I thought, okay, bioinformatics, it's probably more about, software development, developing the computational tools for the computational biologists to use. And then we can use those tools to crunch big data and then do like a more downstream analysis, visualize the data, extract, biological insights. Then that's computational biologist's job. But it turns out people have a very opposite actual opinion for those two. Yeah.
You use the word downstream analysis. If you go to any biology talk, this word downstream research, downstream analysis is used. So can you define what downstream analysis is with an example?
Yeah, that's a great question. Let me take RNA sequencing data analysis as an example. So you know what is RNA sequencing? Essentially, we have like 20,000 genes in the human body, and each gene, it can express or not express this molecule called mRNA. We call the expression of that gene. So you have many copies for some genes, and you have very few numbers for some other genes. That's exactly how the cells, like cell types, are determined, because like all the cells in the whole body, they have the same exact DNA, but we have different tissues. So the tissue-specific, expression of those genes determine the cell type. And RNA sequencing is just a technology to count, for example, how many molecules for each of the genes. And essentially, the raw data will be just like a FASTQ file, is a text file like many gigabytes in size. So essentially just you actually extract your RNA, do some like library prep, and then you sequence them. Then you can actually align those reads back to your reference transcriptome, and you see, okay, this pieces of DNA is from this gene, this pieces of read is from another gene. So you just add those counts up. So you see, okay, how many counts for a particular gene. So you get an idea, okay, how many or how abundant that gene is expressed, right? So you start with this like I don't know, millions of reads, like small reads, like maybe 100 some base pair, but then you need to align that to a transcriptome by some alignment tools, right? Because the transcriptome is huge. Then after you align them, and then you need to actually quantify how many reads in a particular gene, right? And then eventually you get this data matrix. So rows are genes, columns are samples, for example, in bulk RNA sequencing, and the values will be just the number of counts for that gene in that sample. So I'm referring to upstream analysis to the process from FASTQ files, which is like a gigabyte size, to this count matrix, like which is only probably 100 like megabyte, for example, if you have a lot of actually samples, but it could be like several megabytes, for example, if you only have several samples in your experiment. And the downstream analysis will be, okay, then maybe the data analysis, they take over that data matrix, count matrix, which is only several megabytes. And then you do a PCA, then you do differential gene expression and trying to find the pathways that is disturbed between your control versus your treatment. And that's what I call like a downstream analysis. Now, is that clear?
Yeah, I think roughly, but I'll just break down some of the points, right?
Sure.
So when you said alignment, what you're looking at is you're looking at the beginning of the sequence and the end of the sequence. You're
Correct.
lining them up into different samples.
Correct.
And in each samples, you're measuring the count
of
the particular gene.
Correct. You need to align the reads, essentially just DNA sequence, like 100 base pair, ATCG, and then you don't know, because after you sequence, you just don't know where that read is from. And then, but you have the reference genome, which tells you the whole length of that gene, right? And the gene can be like much longer, like maybe like 10 kb long. And then you have this 1 kb, 100 base pair like read. Then you need to kind of match which region that read is from, like across all the genes like in the genome, right?
1 base pair is 1 byte. Is that a standard?
Yeah, 1 base pair is just like one nucleotide. Yes, one letter. If you say ATCG, that's the 4 letters in DNA, and 1 base pair is just 1 letter. Yeah.
Yeah. So in terms of when you say 10K and 1K, so when you say 100 base pairs, roughly how much memory is that? So, or is it like very hard to calculate, or is it like when you said 1K and 10K? So if people have to understand how big these base pairs are,
okay.
So approximately how big are they?
I think a single base is not that big, right? It's just a text file. So think about it. I don't know exactly how you calculate that, but essentially 1K means like 1,000, like 1,000 base pairs. So it's just a sequence, a stream of 1,000 base long, 1,000 letters, I would say.
Got it.
Yeah.
And when you align them, you're ensuring that there are many. Okay, you take the reference gene, and there are many aspects of the reference gene, and you want to align that to a specific gene.
Correct.
And then your sample has to line with that. That's what you mean when you say alignment, correct?
Alignment actually means a line that reads to the reference genome.
Yeah.
And then because you have many reads that can be aligned to a particular gene, then you just sum up all those counts, then you get the number for that particular gene. So that's your expression level for that gene.
And what's the logic of having many samples or like many reads in each comparison?
So for samples, because each sample, because you need to compare, like you have statistical power to compare, for example, what genes expression has changed, right, between your treatment versus control. For example, if you're doing the experiment with just cell lines, and you have like this control, which just you don't do anything, but then you have another set of samples, then you add a drug, for example. Then the cells may grow slower, and then you want to see, okay, what this drug is doing, like for that sample. And then you need actually multiple samples to compare, because you need a statistic power to do that. So you have like at least probably 3 replicates for each condition, so you can get some like stats, right? For example, like you do a t-test, you have to have 3 samples in each group.
Yeah, so t-test would be comparison of means, and you want to be able to show that the mean difference between, let's say, these 3 samples is statistically different to a point wherein the gene is causing an effect that can be measured in the sample.
Exactly. But there's a twist here, because you have 20,000 genes. Like if you just do a t-test, like among these 2 groups, you are going to have a lot of false positives, and that's why like people use FDR, false discovery rate, to adjust for those the raw p-values, because you're doing multiple comparisons here. So this about multiple comparison adjustment will help you to reduce the false discovery. Yeah.
And in one of in the AI technology, I think initially I just wanted to piggyback on one of your point that the way you compare computational biology with bioinformatics, the comparison between statistics and machine learning is something similar, right?
Yes.
And like people confuse, there are a lot of common skills, but the focus is slightly different. There in the statistics, you want to be coming up with an efficient estimator. Machine learning, you're trying to get a better prediction, things like that. There's a slight difference in emphasis, but they're a lot similar.
So, I know. I actually get to know that there are a lot of similarity between the stats and the machine learning, and that's why, like, even like this for the stats people, I got this book, so it's called like statistical learning, right? The people this is actually machine learning, but people refer it to as like statistical learning.
Daniela Witten and Hesti Biani, these are all very famous authors. Witten is Daniela Witten is she's pretty famous. She's the daughter of Ed Witten, the physicist. Some people call Ed Witten the present-day Einstein. That's a viral moment in itself. But that's a very famous book. I've taken a look at that. And, Bob Bishop's Pattern Recognition is another famous book in the same category. These yellow books tend to be important. So whenever there is a yellow book, it's an authoritative book.
Yeah.
So I think for aspiring bioinformatics professionals, who gets into bioinformatics? So if you're a teenager watching this, what kind of skills or what kind of interests would you have that would sort of help you decide that bioinformatics is my thing? I've been looking for all kinds of fields, and think of it as a 17-year-old thinking about different majors in college. And they don't know, probably somebody who is in first year college, they don't know what kind of what kind of fields exist. So what kind of skills, when they don't know all this, what kind of skills match well so that they become a connecting tissue between the interest that they have with the eventual work that they'll end up doing?
Like in my mind, like bioinformatics, you even just look at the word, it's a bio plus informatics. So let's talk about the bio part first. The bio means you're applying the computational skills to biology. So if you really want to be really good at bioinformatics, you need to have a very solid foundation and understanding of how biology works. So if you are like 17 years old, you might want to take some courses like for biology. You should understand what is DNA, what is RNA, how if DNA is transcribed, how RNA is translated into protein, and what are the, like, for example, pathways, like well-studied pathways, like in cancer, like, for example, those biology knowledge is actually critical, right? And for the informatics part, of course, you need to learn programming, like shell scripts, as you mentioned. Like you need to learn Unix commands, because many of the tools that we use nowadays for bioinformatics, they run on command line. And if you don't know how to use Unix, it's just very hard. And also Unix is very powerful and very efficient if you know how to use those commands. Initially, it was actually a little bit intimidating for myself as well when I started like 13 years ago. It's just a black box, and you type something, and then you hit enter, and you execute something, right? Then you repeat. You find it's not that intuitive to begin with, but once you know how to navigate in the file system, learn those like small tricks, it's actually very, very powerful and efficient. And most of the works that we do as a bioinformatician is like wrangling the files, meaning changing the file format from one format to another format, because different bioinformatic tools, they may require a slightly different format. For example, they want first column to be chromosome, the second chromosome column need to be the start, and then second, the third will be the end. But then for the additional columns, they may have different requirements. So you just need to learn how to use this command like cut, sort, unique, this type of thing to clean up the data. So it's very powerful. And of course, learn a programming language like either Python or R or both. I recommend you learn both, because both of them have their own advantages and disadvantages. For R, I still use R most of the time for example, because I do a lot of data visualization, statistical analysis, and R is very powerful for that. And the Python is more like a general programming language. You can use that for like more software engineer, like if you use like Python is better. And of course, like those deep learning libraries that they are all almost like in Python as well. So R is kind of trying to catch up, but it's not as great.
Yeah. And also, like, can you tell me your own evolution into getting to this field? Like when you studied your undergraduate, graduate education, how did you stumble upon, when you were young, you didn't want to become a bioinformatics professional, right? You didn't know about the field. So how did you get to the destination where you are?
Yeah.
So what's the trail of that story?
Yeah, that's a great question. So life is very interesting, Akash, and I never actually, for example, thought about like I would come to the US. For example, I'm from China originally. I came to the States in 2008 to pursue my PhD in a very traditional molecular cancer biology lab. I didn't have any computational skills. I knew nothing. And my under was in biology, but primarily just like, courses and then a little bit of lab work. And at the University of Florida, I started to, learn a little bit more about those advanced like lab technologies, like, do all this like traditional Western blot to look at the protein level and do cell culture, do shRNA mediate the knockdown of a gene expression, and you do all those like assays. So I think the life was good for me. Like I think I published 2 first author papers, like in the 4th year of my PhD, so which was pretty good as a biology major. And then one day, my PhD advisor asked me to analyze this public available sequencing data. I downloaded it. It was 2 gigabytes, I would say, and then it crashed. Like my Excel, I couldn't just open it. So I think that was my first time to realize I lack analytical skills to deal with data. And the biology this subject has evolved to be a very data intensive actually subject. So especially with the sequencing technology, there's just like terabytes data out there, even in the public domain, for you to explore if you know how to, right? The story was I didn't start it just right away to learn by myself. I went to my colleagues, bioinformaticians right there. Can you help me to analyze this data? And then they said, oh, yes. But then I went back to them 1 week later. I'm too busy this week. Can you come back again next time? And a couple of times they keep postponing it. And I was a little bit frustrated. It's like, I need to just do it myself. Like how hard it can be. Like I was asking myself, and then it turned out to be really hard. I would say, because I was the only one actually in the floor, like who was actually learning bioinformatics. Installed like Ubuntu in my Windows machine. I started to learn Unix commands. Start to learn Python and R, like EDX, edX, and also Coursera in the very early time, like 2013. So read a lot of books and watch a lot of YouTube videos. The good thing is, like nowadays, you can essentially find any information that you want on YouTube if you really want to learn. You just need to put in the time. So like during my PhD, like in the day, I still do a lot of actually lab work, but then in the night, I started to just watch the videos, do the homework, and that's how actually I started. And then it took another for roughly 10 years for me to completely convert to a computational biologist or bioinformatician, if you prefer. And I did a computational biology postdoc at MD Anderson Cancer Center, and where I analyze the large-scale terabyte-size genomic data sets, and that's how I started. I think the transition. Yeah.
In the general, that's a great story. I mean, it's sort of inspiring for anybody who's in a different country trying to immigrate to the U.S. and figure out a career pathway in some way. So when people think of, let's say, genomics or genes or anything like that, they think of this company 23andMe.
Yes.
Became very big, which was founded by Google's former girlfriend, Google's co-founder's former girlfriend.
Yeah,
and they had a lot of funding and became like $1 billion company. And then something happened and it didn't take off.
Right.
So that's what people think, because when they think of blood testing company, they think of Theranosys, and they think of genomics company, they think of 23andMe. So what happened in 23andMe? Are there other replacements for 23andMe? So what happened to that in terms of public's perception of genomics in popular culture? 23andMe was the poster child. So can you explain what happened to that so that people have an understanding what this field is more about?
Right. 23andMe, it's more like a company. So they actually send you a kit. So you probably get some of your DNA from saliva, or then they will actually do some panel based SNP array or sequencing, still a panel, a small number of genes, and they will tell you whether you have a risk of any particular disease, or it tells you where are you from, like your ancestry. So for example, so I think they collected a lot of data, but again, those are just small number of genes they are focusing on. And nowadays, because the sequencing is so cheap, you can essentially sequence the whole exon or even the whole genome. There are other actually biotech startups. They provide those type of services, like they can sequence your whole genome under $200, for example.
Oh, because I thought, just to clarify, I thought the whole genome is about $800, and then Illumina and there are a bunch of other players who are trying.
It was $1,000. There was the initial plan, but I believe I saw some company they can provide those sequencing the whole genome even at a lower cost.
Oh, okay.
But I think the power of 23andMe is data, because they also collect phenotype data. So the DNA sequence, what's changing in your DNA, that's called genotype. and that's your genotype data. But they also collect a lot of phenotype data, right? So your demographic information, your gender, age, whether you have any maybe like pre-existing healthy issues, right? So they can do very large scale like correlation like analysis, for example, right? Now with those data, like, okay, if those people have this single nucleotide polymorphism or SNP, like, and that people with that SNP essentially just a variant of that DNA base pair, and most of them have, like, for example, a high risk of heart disease, for example. Like they can do this type of data analysis, which is very powerful. But the other thing is that they only have a limited number of genes. And if you can do whole exome, then now you have 20,000 genes. And even whole genome, you can even have the variants in the non-coding regions of the genome. Yeah.
Okay. And I think that's what people remember in general. I hope there are other competitors in the same field, and probably they didn't figure out how to match, let's say, the phenotypic qualities that they're collecting with the genotypes to a point wherein the prediction accuracy is more valuable than the cost that they were incurring, and the cost went up, and they sort of went out of business. And given this to be the case, I wanted to ask one more biographical question. You're a science communicator. You popularize bioinformatics. You're well known in the LinkedIn community. If someone were to ask, let's say, the present you, how you became who you are, what are the qualities? If you have to trace back your younger self, what will you tell your 22-year-old self or 25-year-old self?
Yes.
Okay, these are the qualities that will become important 10 years afterwards. So learn these qualities. So there might be other people in similar scenarios who are 10, 15 years younger. So what would you tell them that will help them become more like you? If the destination is they want to become like you, so what are the qualities that led you to become who you are?
Yeah, I think no matter who you want to be, like I'm in my like my stage of my career, but if you want to be really successful, I think you really need this self-discipline. I would say that's actually one key thing, and persistence. I think those are two things. I grew up in a very poor family in China. Like the life was tough, but then what it taught me is that you had to actually work really hard. You'd be hungry, hungry because the physical hunger, and that drives me to study hard, because that's the only way to change my destiny. And self-discipline. So if you determined you need to do something, you just need to be persistent and put in the time and work every day. Just to take a little bit more recent example, because you were mentioning I kind of very active on social media, LinkedIn, people know me. It's about work, because I started to post maybe a little over a year ago. So I committed, okay, Tommy, you need to post one post every day for 1,000 days. So I've been posting for 467 something days. So if you do that,
that's phenomenal, actually.
Every day.
Yeah,
Thank you.
Posting it every day is very hard.
So again,
go ahead.
Yeah, again, so you have a plan, then you need to stick to the plan and be consistent, and you will see the results. So focus on your daily goal. I mean, you have a long-term goal, like a year, 3 years, like 1,000 days. But then you break them down into daily goals, right? Maybe yearly goals, quality goals, and then monthly goals and daily goals. The trick is that if you are able to hit the goal daily, like the 10-year goal, it's accomplished in 10 years. That's as simple as that. Yeah.
Yeah, I think there was a famous investor by the name Bill Ackman. He had something similar, wherein like he was going through a difficult phase, wherein all his investments were not going as well, and he
Yeah.
was having like a lot of losses. So what he said was, "I'm not going to focus on the long term. I'm just going to focus on getting better 1% every day.
Every day. Yeah.
Every day, I'll just try to do a little better job than yesterday by 1%." And what
Mm-hmm.
he said was, over a period of 1 year,
Yeah.
that compounding will
Yes.
take care of all the issues. But if you just sit and brood about, oh, that's my 10-year goal, and not take care of the daily things,
Yeah.
it becomes really hard. I think Abraham Lincoln had some quote like this, wherein, what's the point of knowing the North Star if you get, if you drown in the valleys along the way, meaning like when you're going down, if there are
Yes.
some swamps and valleys along the way,
Yeah.
and if you drown there, what's the
Yes.
point of knowing the right direction? I mean,
Yes.
the
Yes.
key is to make sure that you don't drown on a daily basis, and you
Yes.
keep your head up above water, and you
Yes.
keep swimming. And over a period of time, it sort of, you accomplish great things. And this
Ye-
point about
Exactly.
daily discipline is, and you're like the persona of it. And I see you post all the time, and there isn't a single day wherein like I see the post, and there'll be one issue or the other, and that is very hard. I tried doing that, and I
Yes.
just couldn't come up with that topics on a daily basis. So you have to have all that mapped out. So how do you do that? I mean, do you have a
Yeah.
secret sauce?
No, you have to have your own system. Like for me, because I'm doing bioinformatics education, so usually, so there are a couple of ways that I find ideas. The first is like I had a lot of notes in the last 10 years of my bioinformatics journey. So there's a tip actually. You need to document what you did. Write down what you did. And because I have a lot of blog posts, I've been writing blog for over 10 years. And my GitHub repository, I have a lot of notes there. So when I want to write something, I can go there, easily find something. Oh, this problem Tommy had. And then how I did that is, okay, let me just format it as a LinkedIn post, right? So that's one way that I find ideas. The second way is through my daily work, right? For example, I work with a web biologist. They ask a question, and or maybe when I am analyzing data, okay, this thing is like very routine to me, but it could be actually useful for beginners. So what I do, I will actually send an email with a subject line, for example, which so today's actually post is like that. Actually, you will see that which for example, transcription start sites you need to use to analyze this data, because a gene is very complicated biology. So a gene, you can have multiple isoforms. Different isoforms can have different transcription start site, which means like where the genes start to transcribe. And then different isoforms, we call it isoforms, they may have different functions. So it's actually very important to choose which transcription start site for a particular gene. So I just send this subject line, okay, which TSS or transcription start site to use, and send it to my own email. So then in the night, I will just look at those ideas of, oh, tomorrow I'm going to talk about this. I will write it. I use AI, use ChatGPT to reformat it, and then I schedule it. So pre-schedule it. So because I don't really, have time to write it during my day work. Yeah.
I think another tangential question on this very topic, which is very important in today's world where you're inundated with information everywhere.
Yeah.
How do you take notes?
Hmm.
How would you take notes? I mean, because like note taking, some people are of the view that some people still like physical notes. I like to take physical notes of pen and paper and write them in a book. And eventually I'll put them online. So how do you note down stuff?
So for me, my system is very simple. Like there are more complicated people, like they use like different tools, but I just use Google Doc. I will write down. So I think I've been writing for almost 2 years now, daily, just a small blob of what you learned each day, and then what happened each day. Like even just like a diary, like it's like very, very
like a journal.
A journal, exactly. Then just write down. Like for example, if even like I played with kids, I will write it down because those are good memories. And if I read a book, if I listen to a podcast, I will just write down the key takeaways that I learned from those things. It doesn't have to be long. Sometimes I write maybe more words, like 1,000 words, like a day. But then sometimes it will be just like 100 words, 200 words. Doesn't matter. But what I do is that after a whole year, I will feed that into Google's Notebook LM. It's a AI tool. So I will ask questions. Okay, what does Tommy learn during this year? What happened to Tommy this year? So I kind of just relearn the whole year's experience. I find it's very helpful. That's my system. I know for note taking, some other people have different complicated systems. Yeah. But
this is actually quite good. So what you're saying is you'll journal however you want.
Yes.
Just put it all in.
Yes.
And let the LLMs take care of formatting, which it is great at, right?
Yes.
Oh, that's great. I mean, yeah. So you journal and you do it once a month, and you can do summary. What happened last month? What happened a month?
You can do that too. Yes. Yeah.
Yeah. That's quite very interesting. Like you just journal. I mean, I'll probably steal that information and say, okay, I do journaling, but I do journaling for a different thing, for a very specific task. But this is great. Like whatever experience you have, you just write them down.
Exactly.
Personal or otherwise.
We just forget our experience. We forgot, what happened. And sometimes it's like, those are all very valuable experience. Like you learn something, you learn a trick. Like for example, you would mention, for example, YouTube. Like, oh, I learned this trick, how to make the video better, like to reach more people. For example, like how to improve the SEO. Like those little things, you can write it down.
Yeah. And in the current world, where do you see the AI impacting your daily work? Do you think will there be in the popular culture, there is this notion, usually popular culture sometimes gets things right. Most of the times it gets things wrong because it's just, pulling information from the crowd, and which is not the right way to do. If you want to find out something, what is true, you don't want to always be pulling from everybody, right? So one of the notions that exists in this popular culture world is that AI will decrease the number of jobs that exist in the world. So do you see that in your line of work? Are there more people getting hired in your line of work? Are there less people getting hired? Or are they different kinds of jobs that are getting created in your line of work? So what's happening from your vantage point today in June 2025?
June 2020. Yes, definitely. The LLM is great. As I mentioned earlier, like it's good to write. So it's actually 10x your efficiency as a bioinformatician. So for example, if you use VS Code, like you can just integrate with the GitHub Copilot, and you can use different models. It will read all the code in your current directory. It has the context even for your project, and you can just give you a concrete example. The other day, I was using this function in R, but somehow it ran out of memory because the data is like millions of rows and then hundreds of columns. I can just ask, okay, now this function is using too much memory. Tell me how to optimize that. And it will actually automatically just change those things. Okay, you should not store those intermediate variables, which is a big matrix, take a lot of memory. It will tell you why. It will tell you why, right? And then it will rewrite the code for you. I think that's been very helpful for me. But the twist is that you still need to understand it, because after it changed your code, you need to be able to justify it, you need to judge it, right? So whether it's right or not, right? So do some testing. People think, oh, AI probably will replace a bioinformatician's work. I don't think it's likely, at least in a couple of years. Just from my daily work, I still need to communicate with the biologist, who I collaborate with so frequently, because the biology is very, like, with a lot of, exceptions, I would say. It's not like, like computer science, like physics, math. It's a very rigid, like, formulas there. There are a lot of exceptions. For example, like a gene high in this situation may indicate a bad thing, but it could be a good thing in another situation. So you have to just understand the biology. So I think the LLM can help in that sense, but nowadays that people very crazy about the agentic, AI systems. So, okay, how about we create those, like, agentic bioinformaticians, and then they just talk to each other, and then I think the field is developing very fast, but I think it's not there yet to replace us. So my suggestion would be focus on the foundations, know programming skills, right, and then learn biology, collaborate with the biologist, and use AI to help boost your efficiency, but never trust, verify, and everything. That's actually my takeaway.
Yeah, and another analogy that people can think about is, just because we invented cars that go faster than human beings, human beings didn't stop running. Cars obviously go faster than humans running.
Yes. Right.
But that doesn't mean that we stop running altogether. Right?
No.
We still run and we still walk to different places, because we like to walk to different places.
Exactly.
So that's the best analogy. Just because there's a super fast car, you'll not say, "Oh, I won't use my legs anymore." Like, that doesn't make any sense. There are a lot of corner cases wherein you still have to walk to a place, and you can't automate that. I think AI is something similar. It's like
that's a great analogy. Actually, I want to follow on that, because you have a car, but there are places that car cannot get to, right? For example, like there's a small road, and you just want to walk around there, right? So, yeah.
And I think I knew about that analogy before, but when you said biology is all about exceptional cases in the corners, that's what I thought. It's like walking in a very busy street, wherein if you take a car, you'll get jammed. The fastest way is to walk. So human beings will always be important, as long as you figure out a way to walk in narrow spaces wherein you interact with other human beings and stuff like that.
Right. Yeah.
In terms of like our core, let's say, educational module, it's like when people think of RNA sequence, or let's say genomic sequence, what is the core difference? From what I understand, DNA sequences, okay, you have the Human Genome Project, which got initiated in 95 and got over. I think the final stages of it got over in 22 or something, or it got over in 95 as well. So you have this human genome, the universal genome, and you take your blood sample. From that, you get the DNA, which is, let's say, 500 GB, and you compare your DNA with the human genome and see the variations with the human genome and yourself. And you put that in a different file, VSQ file, or people, whatever, call it. And then you use that for analysis. This is what you do in a typical DNA. Now, and this is my cursory understanding. So how would you differentiate this kind of analysis from RNA-seq data, which is essentially gene expression? Like how, from my base level understanding, genes express and genes don't express. And you can take account of their expressions and their non-expressions and analyze that to figure out causality.
Right.
So how would you break it down further?
Yeah, I think I explained exactly like how RNA sequencing work, right? So it's looking at the expression level of that particular gene, right? For DNA sequencing, you're looking at the variants. And think about the difference. This is why you need to understand the biology, because we have two copies of chromosomes. Like for each chromosome, we have two copies. One copy from your mom and one copy from your dad, right? So you have two copies. And then essentially the mutation or the single nucleotide variant, polymorphism, occurs at the DNA level. So that's your blueprint, right, of your genome. And then you mentioned that we have this reference genome project, which sequenced a couple of actually human. And the whole genome is roughly 3 billion
base pairs.
Base pairs, right? And so you extract extract DNA, and then you cannot actually sequence the like a very long DNA. Maybe like there are actually other technology, for example, long read sequence or nanopore. You can sequence like tens of hundreds kb long. But for most of the short read sequencers like the Illumina, they sequence a couple hundred base pairs. So even after you extract the DNA from your genome, from your blood cells, and then you need to chop them into small pieces, right? And then you sequence them, you know the identity, like the ATCG sequences, and then you align that into that 3 billion, reference genome, and then you see, okay, which, and it's not going to, many of them will be like perfect match, but some of them will be not perfect match, but 99%. But that one single nucleotide difference with the reference genome, that's your actually a variant, a SNP, or a single nucleotide and polymorphism. And then essentially you're trying to look at whether your genome is different from other people's genome. The idea of a reference genome is actually, I don't know, not necessarily flawed, because everybody has their unique genome, and 99.9% were identical, but that's like a 0.1% differences make Akash and Tommy.
Yeah.
So that's like how we are trying to do that in the early days. But nowadays we even have the reference genome, like we can have this pan-genome graph. Like it's a different concept, because like the early genome reference is only from, for example, a couple of samples and couple of humans. But now you can sequence, for example, 1,000, for example, healthy human, and you can make a new reference, like they call it the pan-genome. So now, for example, in the case of cancer studies, that's important, because like for cancer patients, you have mutations in your genome. That's why you have cancer. But then you need to figure out, okay, which gene is responsible for the cancer. Then you align your genome to the reference genome. But then you find those like different differences, but you don't know whether that differences, for example, is really because it's really causing the cancer, or it's just a variation like in the population, for example. It doesn't actually do anything. So with the pan genome, actually, you can actually it'll be easier to find those like driver events for your cancer like diagnosis.
Okay.
Yeah.
Okay. And then how would you? So the key thing here is this is the expression and this is the variation data.
Correct.
Yeah.
Yes.
And in terms of people do this concept of normalization in RNA-seq. What does normalization mean? And if you can give, let's say, the intuition of it, so that like people can understand. Well, why do we need to normalize this sequence? Yeah.
Yeah. The intuition would be the first reason is that different samples they are sequenced in different depth, which means how many total reads are sequenced for that sample. For example, if you have 2 samples, sample 1 you sequence like total you have 10 million reads, and for like sample 2 you have like 2 million reads total. Then if you look at a specific gene A, in sample 1 you have 10 counts, and then in sample 2 you have 20 counts. Like
yeah.
But then that's just simply because you sequence deeper,
Yeah.
So then you need to somehow normalize it to the total library size. This is like a first intuition, like how to normalize the data. But there are other issues, like genes with high expression, they are more variable because it's like a binomial, is a negative binomial actually distribution.
Okay.
So the bigger the mean, the bigger the variance, and then you have some actually some statistical method to normalize that as well. Yeah.
Okay. Okay.
Yeah.
And are they like clinical scenarios where DNA sequence is more influential than RNA sequence? And what are those scenarios? Like, if you can think of them.
DNA versus RNA. So it really
Yeah,
depends.
yeah, where RNA is more clinically significant in informing the clinician or the physician to diagnose some disease condition.
So RNA is more important. So it really depends on the biological conditions or questions at hand. And some, for example, some genes, like in cancer for diagnosis, like some genes, because they're mutated, there's well-known oncogenes, they're mutated, or tumor suppressor genes, they are mutated. So if you find variants, a DNA mutation, then that's probably the reason. But on the other hand, there are some actually genes, they don't change at the DNA level. Instead, they increase their expression level. For some oncogenes, they don't change the DNA, but then they have just a lot more copies of the mRNA that drives the oncogenic events as well. So it really depends case by case. Yeah.
And is there like it depends on case by case, and there is no universal blank statement. So what I mean is, if I say something like BRCA1,
Mm-hmm.
so it's not a DNA count being messenger RNA count being higher. It can be
Yeah.
either or, right?
Yeah. It can be mutation, right? It can be also the expression level, and it's no actually BRCA1 is its promoter is DNA methylated in some of the cases. That's why the expression level is low. So it can be expression level, but it can be also maybe mutation level.
And what are the limitations of this technology, RNA-seq count, for finding? Ultimately, all we're talking about it from the standpoint of a layperson understanding this technology
Yes.
as to how it will influence their physicians making their diagnosis. So what are some of the limitations of this?
The limitations of RNA sequencing. RNA sequencing has been around for many years now. It's very popular. It's very easy. And now it's also very cheap to run RNA sequencing experiment. I think the sequencing is like about like 100 something or $200. The limitations, I would say you need to use the right tool to address the right question. So RNA expression, you can only look at the RNA, the expression levels, right? And if that disease is caused by mutation only, but not expression, you cannot detect that with your RNA-seq. So that's why, like, to everybody with a hammer, everything's a nail. So you should actually you should use the right tools for the right questions. Yeah. Again, you need to understand the biological system, understand the domain knowledge behind the disease, and use the right tool. And I would say everything has its limitations, but it also has its own actually advantages. So just need to figure out which tool to use in which condition. Yeah.
Yeah, and also slightly biographical tilt. You did some time in academia and some in research labs, some in the corporation. So how do you find the lifestyle in terms of your day-to-day work? So academia, a research lab, and an industry lab.
Yeah. So I'm in the R&D group, right, in a pharma company, research and development. So I still do research in that sense, and I would say it's very similar in terms of my day-to-day work. It's analyze the data, communicating, making decks, do all this like data wrangling and visualization, statistical analysis. It's similar. So the one thing that is different is that in academia, it's more about papers, or because you need to graduate, you need to focus on paper. If you need to apply for grants, you need paper. And it's very, let's say, competitive, like in terms of a specific project. Okay, this is my project, and I am the first author. I think that in academia, there's the mindset I used to have. Most of us used to have, but in a company, in industry, biotech or pharma, the goal is a little bit different. Publication is good to have, but really the goal is to work together, collaborating with each other, and let's do it together to make a drug and make a medicine to benefit patients. So it's a different goal, right? And it's more collaborative, I would say, in industry. That's the difference, I would say. But in terms of day-to-day work, it's very similar.
Yeah. Okay. So because you're a research scientist still, so your core toolkit, so to speak, hasn't changed.
Correct. Yeah. Communication's important, right? No matter where you go. Yeah.
And how much is communication? I mean, piggybacking on that point, communication is important regardless of which field you're in.
Yes.
And especially it is a very important, collaborative setup wherein you're sharing attribution. So if I have to summarize, in academia, the doing collaboration is fine, but how do you share who gets credit and who gets how much credit?
Yes.
Right? In industry, it's more about getting the work done to a point where the eventual goal is satisfied.
Yes.
So communication is very important in all industries. So how important is it in biotech, where it's a very social setting? You talk to a lot of people with different backgrounds. So how important is it?
Yeah, it's important, right? When you, for example, bioinformaticians, we talk to the wet lab, like, who generated the data. I think a good bioinformatician needs to stay close with them because they generate data. They know the nuances of the data. If you don't ask, they probably will not tell you, because if you find something that is weird, for example, even from your PCA analysis, they may actually tell you, oh, maybe those two samples actually maybe get mixed up somehow. Like, we have some notes here, and then you find that you get confirmed with your analysis. And maybe they will say, oh, this batch of experiment, I know the cells are not healthy, and then maybe the quality is not as good. So you just need to, communicate with them to know the nuances of the experimental design, how they generate the data, right? And then on the other side, after you finish the analysis, you make a deck. Try to communicate with them the results, like guide them, because your job is to give insights and help them to make decisions and help them to understand the mechanism of action of your drug. So you need to communicate really succinctly and to them, okay, this is what I found, this is what I did. And also keep in mind their background, right? They're not like you, and they're not statisticians. Explain things to them in a very simple way. That's very important, right? That's just for collaboration, but also in terms of like stakeholder management. When you present to people who are at higher management, they don't need to know all this nitty-gritty details, like how you analyze data, how you fix this bug. Then you have to actually communicate really well what's the key message from this analysis, like maybe even just 1 or 2 slides from all your hundreds of decks. So I think communication is important, and to get through the importance of your analysis, and also catching the errors, potential errors in your analysis just by talking to the biologist. Yeah, the communication is important.
Yeah, and in terms of how data sets, how open are data sets in biology? And can you tell a layperson some of the sources to find some of the data sets that they can work on? Let's say somebody is a Python programmer who works on data science, but doesn't know much about biology, but is interested. Once in a while, he hears some keywords, and he's sort of curious. And he's also curious because there are the data quantity is like GBs of data, and he's not used to that. Like most of the people who are statisticians, they're used to, let's say, KBs of data that they put it on an Excel sheet or SPSS or Stata or any such tool. But they're working on smaller data sets, but they have the skill sets. They probably know some scripting. So if they have to get started because they have the skill set in terms of the toolkits, but they don't know where the sources are, what sources would you recommend where there are open data sets to get started with?
Yeah, I think the one that I would recommend is go to GEO, like it's from NCBI. And people, after they publish their paper, they usually upload to this data repository. and then you can just search like whatever topics that you're interested in. Okay, RNA sequencing, or then okay in such cancer, and it will tell you the data sets that are available there. Then you can really actually download and use those data and to play with. Yeah.
And is that the go-to source? And there are no other sources in terms of we have PD. I was thinking PDB, protein database.
Oh, PDB. Yeah, it really depends on what. Yeah, PDB. Like if you are interested in like protein structure, yes, PDB is the one to go. But for like expression data, for like protein binding and like ChIP-seq, like DNA methylation data, like mutation data, whole exome, those are available actually in Geo. Yeah.
And since last year, again, this is what is popularly known, right? Chemistry prize was given to Demis Hassabis, David Baker, and John Jumper for their work in protein structure and protein language models. So how did that occurrence affect your field? Like you suddenly realize, oh, these people are doing some interesting stuff. Do you think like knowing protein structure would have impact in your daily life if you do something like that?
Like I'm not the chemoinformatician, like which means I don't do this. For example, docking stuff. For example, you have a small molecule. You try to see okay where does it bind to the protein, and the PDB and all the database and also this AlphaFold thing. They can help in that sense. So you maybe speed up the the screening of your small molecules, and even you can do virtual screening. Okay, see which molecule can bind the protein, inhibit the enzymatic domain, for example. Then you kind of synthesize those small molecules. It can potentially speed up the drug development process. Yeah, but yeah.
And how do you stay updated about your own field? Like, well, where do you get your information? I'm assuming you're constantly reading and journaling all the things that you learn. What are your sources? I mean, you don't have to give all the sources, but at least the kind of sources that you would recommend other people to sort of bake into their appetite, so to speak, so that okay, they can add it as part of their reading list and they'll start
I follow people on social media, Twitter, LinkedIn, like they're bioinformaticians, they're professors, and you can also follow those journals, like Bioinformatics is a journal. You can follow them in bioarchive papers, and you can follow even genobiology, cancer cell, like those journals in the bioinformatics field, right? So follow them, the labs. And also, you can subscribe to those journals. When they publish a new paper, they will send you a notification, and you say, okay, there's new tools there. Yeah, so I think those are two my primary resources.
Yeah, I've seen academia, especially in the US. It's not so true in other countries. But if you're in academia in the US, academic Twitter is very active. In the sense, like anybody who publishes anything, they're always posting it on Twitter. And this is long before X became what it is right now. I mean, even in 10 years ago, in 2015, academic Twitter was very active.
Yes.
Like in different fields, people in math would publish and put their archive links on Twitter so that things became popular. So is that also the case in biology and bioinformatics?
Yeah, it was very popular, right? I started my Twitter over 10 years ago, and I follow a bunch of people. But recently, after Elon took over Twitter, I know a lot of people, academic people, they moved to Blue Sky, a different platform. So I have an account there as well, follow a bunch of people. So just follow where the crowd go.
Yeah. What is that one misconception about bioinformatics that general people have that you can sort of dispel, or you have like one or two misconceptions people might have?
One of the misconceptions is that doing analysis is very easy. Like just press a button, then you generate the figures. Like that's one key misconception. But in reality, there's so many things you have to do. Like even before you analyze data, you need to install a tool. It can take you hours. You need to make a decision which reference genome to use, and and then you get the data, or you have to do exploratory data analysis. Okay, find whether there are any mislabels of the samples, do enough quality control. And then eventually you get to do the real work and then then you have to clean up the data, making plots and do statistical tests. Yeah, maybe that's only like 10 to 20% of your work, but most of the time you spend on other other stuff.
Setting up the actual problem. So it's like everybody thinks when there is a movie called Good Will Hunting, it's about people doing math on chalkboard.
Yes.
Right? So people thought like everybody who's a researcher is always writing things on the chalkboard. Yeah. Oh, that's the eventual stage. You have to do a lot of work to collect information, figure out, do literature survey, read other people's work. It's not always some tree structure and you draw lines between the nodes. Right, it's not as simple as that.
No.
Yeah. And are there any other misconceptions apart from the fact that it's not easy?
Other misconception. I'm not sure whether it's a misconception, but I've seen a trend that there are actually tech people. They're trying to build a biotech company from a pure computer science background, and they think they can just solve like biology with brute force, like computational methods. I don't think it's likely. So if your company is for drug development, you got to have an expert domain knowledge in drug development in biology. And that's the combination of the computation and the biology which makes the company successful. There's another thing that I want to say.
I think that's been explored in every field. Like computer scientists think they can get into any field and take over, which is not the case. Every time they try to take over, something bad happens in the world, and then they realize you blame the nerd who did it.
Yeah.
That being said, it's not to be overlooked. No field is easy, and there are intricacies. So, do you think cost of drug discovery? This is something that we didn't probably plan out, but do you think the cost of drug discovery is going to have a significant change, or is it still in the cost of drug discovery would mean budgeting, right? It's still so does it still take 10 billion, 10 years for one molecule to come through? Or would you think with the advent of AI and bioinformatics coming together?
I think it should help. I just don't know how transformative it can be, because the bottleneck for drug development is still the clinical trials. You just take multi-year phase 1, phase 2, phase 3 to get a drug and then approved, right? It just takes that long. But of course, AI can help, patient stratification, maybe site coordination to recruit patients. I hope now it takes 10 years, maybe it reduced to 8 years. That's still a very good, actually, progress. I'm actually optimistic in that sense, but I don't think you can say, okay, take only 2-3 years to make it.
Yeah. So people who say that it'll automatically be automated, wherein it'll happen in 2-3 years. The way I push back is my example of just because AI was invented, the voting that takes place every 4 years in the US, people don't start voting every second. Even if they vote every second, it doesn't mean that more people vote over a period of time. A lot of people don't even vote. That behavior hasn't changed. So some things remain constant no matter what. So whenever I say this data point, if you look at voting patterns, just because there's new technology and AI, it doesn't mean that a lot of people who are supposed to vote end up voting. A lot of people just stay back home because they think they're not into it. Thanks, Tommy, for all the answers that you gave. And thanks for taking the time out and doing this podcast. And hopefully you'll continue to do this education bit on social media. Thanks a ton.
Thank you very much, Akash. I really enjoyed the conversation. Thanks for having me.
Yeah. Take care, guys.